all. This is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. So today we have a very interesting study. This is from the Harvard Medical School, Boston University, and Dana-Farber Cancer Institute. The researchers have created an intervention, a Decoy ACE2 receptor to bind with the SARS-CoV-2. And it has shown great efficacy in vivo in Syrian hamster models. While there have been previous studies for the ACE2 enzymes to be injected to bind with the SARS-CoV-2, and these studies have not been very successful, this is the first one that has actually shown how to create a successful intervention of decoy ACE2s. So let's look at this. And I would actually, it is so funny that in the beginning of this pandemic, Dr. Vasaris Alexandros from Greece had commented once in our discussions that isn't this possible to create ACE2 decoy that can connect with SARS-CoV-2. So here we are. Beautiful study, great work by the researchers. Let's look at this study together. So first of all, this is Drbeen.com. In the description of this video, there is a link to Drbeen.com, 900 more medical lecture videos for only $67. This is only for the holidays, and this is a gift for you. And this is not a monthly recurring price. This is just a one-time payment. This is the study. Optimized ACE2 decoys neutralize antibody-resistant SARS-CoV-2 variants through functional receptor mimicry and treat infection in vivo. We'll talk about this. And there are some more reference links in the description of this video, some that discuss what is colectrin. We'll talk about that. Here is another about colectrin, and then structural bases with the colectrins in here as well. So with this, let's look at my diagrams. So this is the spike protein. It is sitting there and being very angry because I think it does not like our research workers. So these are gifts for humanity, and they're continuing. Let's look at the abstract of the study before we continue to some details, not all of it. For all of it, please make sure that you read it as well. Abstract. As severe acute respiratory syndrome coronavirus SARS-CoV-2 evolves to escape natural antibodies, natural meaning being produced by the infection, it also loses sensitivity to therapeutic antibody drugs. So the monoclonals that we've made, it is actually escaping them as well. By contrast, evolution selects for binding to ACE2. So as much evolution SARS-CoV-2 is doing, it still has to make sure that it is able to bind to ACE2. Correct? So evolution has to make sure that that area, the receptor binding domain of the spike protein, continues to become or stay conserved to be able to bind to ACE2. And so that is their key that they would use in the study. The cell surface receptor required for SARS-CoV-2, that is the ACE2. Consistent with this, we find that an ACE2 decoy neutralized antibody-resistant variants, including Omicron, with no loss in potency. An ACE2 decoy. This is the first time this study is showing it. Previous studies failed or did not show as great a benefit. So ACE2 decoy neutralizes antibody resistant. So that variant that has become resistant to antibodies is still caught by the ACE2 decoy and with no loss in potency. To identify design features necessary for in vivo activity, we compare several enzymatically inactive. So that means these ACE2 that they made as an intervention, These would not go into our body and start working as ACE2 and start picking up angiotensin 2 and converting that to angiotensin 1 to 7. And all of a sudden, we have a different kind of a problem. That's not going to happen because these ACE2 are modified to make them enzymatically inactive. And they are attached on the antibodies. That is how many interventions are. So the Fc effector is silenced. So the antibodies fraction constant has biological activities, so that part is silenced. It does not do its activity. Or, for example, activating complement and causing inflammation. So Fc effector silenced ACE2 Fc decoys. Inclusion of the ACE2 colectorin-like domain not only improves affinity for the S protein, but also unexpectedly extends serum half-life and is necessary to reduce disease severity and viral titer in Syrian hamsters. So this colectorin-like domain, and I'll explain that a little later, was important in this study. They found it to be important. In previous studies, this one protein part was actually removed when creating the ACE2 decoys. And so researchers actually found out that this was important to have. Fc effector function is not required, and we'll discuss a little more later. The activity of the ACE2 decoy receptor is due in part to their ability to trigger an irreversible structural change in viral S protein. Our studies provide a new understanding of how ACE2 decoy function and support their development as therapeutics to treat ACE2 dependent coronaviruses. Beautiful. So I want to go back here for a second, and I want to show you this little spike protein. So do you see that these little 3 legs over here? So this is a post-fusion stable spike protein. That is a state of a spike protein once it has bound to ACE2. So that is what they were alluding to, and I'll explain it a little later. Just keep this in your mind. Good study. So here is the discussion of it. So again, a high-level overview. So normally what happens is we have a SARS-CoV-2. SARS-CoV-2 has spike proteins on it. We know that. The whole world knows that now. These spike proteins here, they allow this SARS-CoV-2 to bind with ACE2, which is an enzyme on our cell surfaces. Again, we all know it. I'm just kind of bringing us back to the same page. Now, what happens is the humoral immunity, that is the antibodies that we produce. So that we produce with the natural infection or with the vaccine. Again, this study does not talk about vaccine. They talk about antibodies produced. So when we have antibodies, these antibodies will bind with the spike proteins of the virus, which then puts pressure on the selection pressure on the spike on the virus to create mutations that will try to escape the antibodies. And we have done this discussion before. The virus doesn't have any intelligence to say, I need to change because an antibody is binding to me. What virus does is that as it is being built again and again, the daughters are being formed in our cells. There are lots of kinds of mutated daughters. That is just natural. There are some daughters who will make spike proteins that are just totally trash. Those spike proteins don't work. They are like disabled spike proteins. And so these daughters will not go anywhere. They would just not be able to bind to ACE2. These viruses would die or be destroyed. Then there are going to be viruses or daughters that would have their spike protein very similar to their parent. So let's call them almost parent. And then there would be other spike protein variations, and some of them will be more fit than their parent. And because they will be more fit, they will be able to outcompete their parent and get into the cells, and those daughters will then replicate more, and they would make more viruses, and the person would shed them more as well, and so they would get a chance to spread. So that is really what happens in selection pressure, and not that the virus sits down somewhere and says, you know what, I need to change here and here and here. So anyways, as we try to bind with the virus, our antibodies, the virus mutations, those mutations that will survive these antibodies will start surviving in that person. And when that virus will be shed from that person, now we have a variant that is out in the field. So here if you see, when the variations occur, the variants kind of escape the current antibody learning that our immune system has. So for example, if I was infected with alpha, Omicron can escape alpha antibodies or antibodies to alpha, right? So that is the normal behavior of the variant. And if you read here, the researchers are saying, consistent with this, the beta and delta SARS-CoV-2 variants demonstrated resistance to some antibodies developed for COVID-19 treatment and prophylaxis, so the ones that are monoclonal. The subsequent Omicron BA.1 variant was resistant to almost all of these antibodies, including casirivimab, imdevimab, etesivimab, bamlanivimab, tixagevimab, silgavimab, ADG10, adintrevimab, ADG20, and ADG30. So the more recent Omicron BA2, BA3, BA4, BA5 variants are also resistant to these antibodies as well as to sotrovimab. The FDA has therefore placed restriction on the use of each of these antibodies, and we have done this discussion before. There is also concern that SARS-CoV-2 may eventually develop resistance to Paxlovid, the oral antiviral that was most effective in trials and is most widely used for treatment of COVID-19. The point they're making is that whatever are the interventions that we have, monoclonal antibodies or even Paxlovid, these are slowly becoming escaped. So, what did they do? What they did was, let's just very quickly look at a high-level diagram of ACE2. So if those researchers are seeing this video in their honor, I'm going to show the actual diagram from their here. So I'll show this as well. So here is the ACE2 with PD, and this is ACE2 CLD. So in your honor of the researchers, here are the actual diagrams, and here is my depiction of it. So an ACE2 enzyme is made up of multiple parts, but important parts here in this context. One is the PD part, or the peptidase domain, and the other one is CLD part, or colectrin-like domain. This CLD part has a tiny little helix, or another part of the protein that can go across our cell membrane, and this is how the ACE2 is anchored in our cell membrane, plus the second messenger system works through this mechanism. So please keep in mind that we have a PD, or peptidase like domain, or O domain. This PD has the area where the SARS-CoV-2 spike protein binds. And what part of the spike protein binds there? We know it, receptor binding domain binds there. Now, if you see here once more, these are two ACE2s that have dimerized, or that have come together. This is called homodimerization, or coupling of two identical proteins to make a new protein complex. So these are the two. And now what happens is, the spike protein, if you see here, this spike protein has these little black areas on it. These black areas are depicting, or are my depiction of the of the changes in the spike protein near the receptor binding domain area, or S1 part, to evade and escape the antibodies and monoclonal antibodies and so on. However, the SARS-CoV-2 spike protein has to keep its receptor binding domain conserved enough, consistent enough that it can keep binding with ACE2. Because if it cannot bind with ACE2, if there is an accidental mutation in there, then that daughter cannot bind with another ACE2 to enter that cell, and that daughter is gone. So back here, this is a dimerized ACE2. This is the helix I was talking about, and the CLD, the collectrin-like domain, that collectrin-like domain has a job of creating this dimer. So enough, we have PD and CLD. Good. Now the researchers had this hypothesis that if they made an ACE2 decoy, an artificial ACE2 from outside, then that can neutralize the SARS-CoV-2 despite the mutations in the SARS-CoV-2 spike protein. And their reasoning was the following. They said the receptor binding domain has to stay consistent. Receptor binding domain of the spike protein of the virus, right? It has to stay consistent. It has to stay conserved so that it can bind with ACE2. And if it changes the receptor binding domain, then it will not be able to bind to ACE2. So imagine this intervention, this drug, if you will, that these researchers have made. If the virus tries to mutate away from it, then the virus will have to change its RBD. And as soon as virus changes that virus is not going to survive. So this is why I wrote here win-win. So this is the Decoy ACE2. I didn't make the helix, so here it is. This is the Decoy ACE2. And it keeps binding with the SARS-CoV-2 spike protein regardless of the mutations in the SARS-CoV-2. Because SARS-CoV-2 has to stay fit to bind with ACE2. And if SARS-CoV-2 changes its receptor binding domain to try to be clever to escape this drug, then this SARS-CoV-2 is not going to be able to bind with another ACE2 and done. It's not going to be able to work. So it is a win for us and win for us either way. So researchers said that in the previous studies that tried to create these decoys, they did not work very well. And the reason for that, and this is my words, didn't work very well. They had expressed it differently. It didn't work very well because what these studies did was, if you see here, they had changed the surface of the SARS ACE2 to fit the spike protein of a specific variant. So it would work very well for that variant, but as soon as the variant has a change, this ACE2 enzyme cannot bind with it. So that is how these previous decoys didn't work. So these researchers, what they did was they said, you know what, we'll keep this ACE2 enzyme surface the same as is in the humans. We'll not change that. So that the only thing they did was they made sure it is enzymatically inactive, number one. It stays standard. Plus, in the previous studies, if you see here, previous studies did not have the CLD. They thought the collectrin-like domain part is not necessary. So they removed it. Researchers here, they actually created multiple kind of these interventions. Some had CLD and some did not. So the one that had CLD became successful, and so they discovered that CLD is very important. So here, in one of their flavors of these decoys, they had the CLD left in place as well. And then, of course, there is this Fc part of the antibody, and that Fc part is silenced, meaning it doesn't do its biological actions, and I hope it will also not bind with the Fc receptors on, for example, natural killer cells or macrophages or other cells. So those reactions will also not occur, which is a good thing. That also means that researchers are not looking for macrophages or other cells to pick up this bound virus through their Fc receptors and eat them up and phagocytose and destroy them. So anyway, so that is the structure they built, or this built 4 structures. This is the one that became the most successful. So their finding, what they did was they built this structure and they built another 3, and they then infected Syrian hamster mice with SARS-CoV-2. Then they treated them with these decoys. So all other decoys failed, but this decoy became very successful. So let me actually show you the researchers' statement about it, and then we'll go back to the explanation. So here they said, we compared the in vivo antiviral activities of the 4 ACE2 Fc compounds in a rodent model of SARS-CoV-2 infection. So they had 4 flavors. When challenged intranasally with SARS-CoV-2, Syrian hamster demonstrated rapid weight loss, high viral titers in nasal turbinates, so nasal areas, and lung, and severe lung pathology. We compared the ability of the 4 ACE2 Fc proteins to alter the disease course in this model by administering a total of 40 mg per kilogram per day of the ACE2 Fc or vehicle, let's call it placebo, for 3 days beginning 8 hours before intranasal challenge, with 1 multiplied with 10 to the power 4 plaque forming units of the Wuhan isolate of SARS-CoV-2. All animals were euthanized on day 3. We determined body weight loss and obtained viral titers from homogenates of the nasal turbinates and lungs. What did they find? PD, CLD, FC. So that is what I showed you. ACE2 with the PD and CLD, collectin-like domain, and the FC part, so this 3-structured protein, was the only compound to attenuate disease severity as measured by weight loss. Treatment with PD-CLD-FC resulted in a statistically significant reduction of approximately 100-fold in nasal turbinate viral titers and 10-fold in combined cranial and caudal lung titers. Wow, beautiful. None of the other compounds resulted in a statistically significant reduction in nasal turbinate or lung viral titer. So they had 4 compounds and one of them worked. So it was not a fluke. It's not that the previous ones didn't work and this one, they just figured out it will work. They actually had 4 as well, and one of them got discovered to be working very well. So back here. So this is what they had. Their PD, CLD, FC model binds to the S proteins of all variants because this part of the spike protein, the RBD part, is conserved, majority conserved. If you go down to RBM, I'm sure it is even more conserved. So standard PD of ACE2, CLD included, and they found that collectin-like domain enhances apparent affinity for S protein. So this affinity to bind with S protein becomes enhanced apparently when the CLD is present. And we know, we have done these discussions of conformational changes where one protein's amino acid and charges can influence other parts of the protein as well and can modify its even physical shape of it. Of course, that physical shape would then result into structural change plus the ionic changes as well. Improved pharmacokinetics of the decoy. So the decoy stayed in the serum for longer period of time, which is also excellent. Binding to the decoy. So here, this was the funniest. I was chuckling when I read this. So here is a normal spike protein of a SARS-CoV-2. Imagine there is a SARS-CoV-2 attached to this thing as well, and there are many spike proteins, right? So here is a spike protein of the SARS-CoV-2, and that encounters this drug, this ACE2 decoy. And not only the ACE2 can bind with it, it actually opens it up. Remember, the spike protein is in a pre-fusion state and then post-fusion state once it is bound. So what happens is, again, I should have removed the S1 and then open up the S2 part. So you know the anatomy of it. So what happens is, we have done this study discussions in the past that ACE2, when the spike protein reaches, so if you can look at my hand, when the spike protein reaches near ACE2, then the electromagnetic forces cause the spike protein to bloom, to open up. And the S1 part is separated, and TMPRSS2 comes in and separates that, cleaves it, S1 removes, and then the S2 kind of opens up. And then, S1 opens up and binds with this ACE2, and the S2 then fuses with the cell membrane. So we've done these discussions many times. Here, the important thing is the spike protein enters irreversible post-fusion state. And when it is in that state, it cannot bind with another ACE2 anymore. It is a fired gun. It cannot fire again. So it is so funny that this intervention blooms it. So it just keeps touching them and they bloom. I'm just making up a story now. When it combines with it, it goes into post-fusion state. It's just funny for me. So what was the structure itself? So there was this antibody they produced. In that antibody, the binding areas were the PD and CLD. The FC part was modified as well to make it silenced. So they said that FC part enhances the avidity of the binding, and the CLD and PD were, CLD included, caused enhancement of affinity. And so if you see here, this is one of the charts that they have, and I wanted you to see here, this is their ACE2 FC. And if you see, all other drugs failed for IC50 test or failed to help with the infection, while this drug did not. So that is a lots of fun research. So this is the discussion today. What I do not know is when will this become available, or if it will become available as an intervention, but they have proven from their research that this is a good intervention that is potent, that doesn't lose potency, is against almost all kind of variants for SARS-CoV-2, and I think is something that we've been talking about for years now. So let's see if it becomes an intervention or not. Thank you very much. And once again, please like, subscribe, and share. If you like this work, and if you would like to support this work, there are links in the description. You can buy me a coffee, you can use PayPal, you can become a patron, and so on. Thank you very much, and I'll see you tomorrow.